Hello all. In this session we are going to see the introduction to the ANOVA, that is nothing but the analysis of variance, and this technique is widely used in the different fields of research area such as economics, biology, education, sociology, etc. This technique is going to be used only when if it is having more than 3 samples which are available in the case, and this was first used by the K.A. Fisher. So before starting this, I am going to revise some parts related with the hypothesis selection and hypothesis testing things. So in that, the hypothesis is nothing but the assumptions which are going to be made by the researcher. These are going to be classified into the two different types of hypothesis, which are nothing but the one is the null hypothesis and the another is alternate hypothesis. There are two different conditions for the acceptance or rejections of hypothesis from these two, which is nothing but when we are going to accepting the null hypothesis, at the same time we should have to reject the null hypothesis. In the second case, if we are rejecting the null hypothesis, it means that we should have to accept the alternate hypothesis. This is going to be defined or considered in between the two different types of errors, type 1 error and type 2 error. Now in this table it shows that when the null hypothesis is true, it means that we should have to accept the null hypothesis. So if you are accepting the null hypothesis, then it is the correct decision. If null hypothesis is true but we are rejecting the null hypothesis, then that error is called as a type 1 error. In the second case, if null hypothesis is false, and in that case if the null hypothesis is false, is wrong, then it means that we have to reject the null hypothesis. So when we are rejecting the null hypothesis, then it is called as a correct decision. If we are accepting the null hypothesis, then it is nothing but the type 2 error, which is also called as a beta error. Next, there are some common tests which are going to be used in statistics. The first one is nothing but the Z test. This test is used when the sample size is greater than 30. So in that case we are going to using the Z test. Second case, the T test is going to be used when the sample size is less than or equal to 30. In the second case, the population standard which is given in the case, which is population standard deviation, is not known, then in that case also we are using the t-test. The next one is very important, which is nothing but the chi-square test. This test is used to test the goodness of fit for that particular case study or that for that particular process. Second one is the test of independence, the independency of number of variables which are present in the data. On the basis of that data, we are going to using the chi-square test to find out the independency of each and every variable with each other. The last one is very important, which is nothing but the F test. The test is developed by the K.A. Fisher. This test is used to equality of variances of more than two population is known as analysis of variance. Next one, now we will start with the actual analysis of variance technique. Now what the basic principle ANOVA is says that when there is having three population means, let x, y, z are nothing but the population means which is available. Now when we are using these populations means, the data, what we have to do in that by using the ANOVA, here we have to examining the amount of variation in between these three population means relative to the amount of variation between the samples. We have to find out the amount of variation in between these three population means as well as we have to find out the population mean or relative to the amount of variation between the samples. And after that we have to estimate of population variance. Now this variance is going to be considered in between the sample variations or in between the sample variance or within the sample variance. So there are two different things that we have to consider when we are solving the problem by using the ANOVA. So here we will clear that when we are having the more than three variables, more than three samples, then in that case only we are going to using the ANOVA technique. Here when you're defining this principle, when you're examining the variations, so at that time we have to consider the F test and whatever the results which are coming from this data is nothing but the F test, and that F value will come from this analysis. Next, let's have a simple example to understand ANOVA. Now we have to define first the null hypothesis as well as the alternate hypothesis. So let's have HO is nothing but the null hypothesis and HA is nothing but the null alternate hypothesis. Here when HO is equal to mu1 is equal to mu2 is equal to mu3 and so on up to the mu k for the k value or the for the k variables for the k population, and where HA is nothing but HA is going to be considered only when if at least one population mean is different from the others. Now here let's have a simple example. We are having the three different institutes. Institute 1, Institute 2, Institute 3. The Institute 1 is having the 1000 number of student. Let's have a 2000 number of students in the Institute 1. In the second institute we are having again the 2000 student, and Institute 3 is consist of more than that 2000. Let's have a 2200 of students. Now here from these institutes what we have to do, let's the mean population of Institute 1 is mu1 and the mean population of Institute 2 is mu2 and the mean population of Institute 3 is mu3. Now it's very difficult to consider all the 2000 student data to analyze that particular variations. Now let's have a consider any one subject for all the three institute, and for that particular subject we are going to we are considering the results which are going to be offered by the student, and that results are going to be considered. Let's have for the mathematics subject, and for this mathematics subject we are considering the data of all the students to analyze it. So it's very difficult and time consuming process when you're considering the 2000 samples or the 2000 numbers which are coming from the 2000 student. So what we will do now, let's have a consider the sample of 10 students from the Institute 1. Now that 10 samples is having some value. These values are nothing but the values which are going to be considered, or the marks which are obtained by the student. Similar way, we will find out from these 10 samples, we will find out the mean of the population for the 10 number of student. Let's have an x1 bar. So x1 bar shows the mean population of Institute 1. Second case, again we are considering the same strength of the student as a sample, and again we are going to calculating the mean population, which is nothing but the x2 bar. Next one, for the again third institute, we are considering the sample of 10 student, and we are calculating the mean of samples, which is nothing but the mean population of these 10 students. So x1 bar, x2 bar, x3 bar are nothing but the mean populations of sample 10 from the Institute 1 2 and 3 respectively. So if x1 bar is equal to mu 1, when we are defining the null hypothesis, or when we are considering the null hypothesis, or we have to prove the null hypothesis, it means that x1 bar should equal to x2 bar should equal to the x3 bar. Then only we are going to accepting the null hypothesis. If any one from these three mean population, let's x2 bar is different from the x1 bar, and x2 bar is also different from the x3 bar, then it means that we have to reject the null hypothesis. And what we have to do, we have to accept the alternate hypothesis. So it's very important when you're defining the null hypothesis and when you're defining the alternate hypothesis. So in that case, in that conditions, we should have to remember that how we will define it and how we will select from the data. So hope you understand at what conditions we will accept the null hypothesis and at what conditions we will accept the alternate hypothesis. In the next session, we will see the number of different steps which are going to be involved in the ANOVA technique. Okay, thank you all of you.